Welcome back to the Pre-Vetted podcast, where we spotlight extraordinary people and remarkable talent reshaping our world. Today, we are joined by Ewelina Kurtys. She's a scientist turned entrepreneur. She has a PhD in neuroscience and more than 20 peer-reviewed papers. After her academic career, she moved into technology commercialization, advising deep tech companies on sales, partnerships, and strategy. She's currently a strategic advisor at FinalSpark, where she applies neuroscience expertise to build next-generation biocomputers from living neurons. She's also founded ventures like Sync, mental wellness app, and Ekai.io, a business development consultancy. She has experience spanning research, entrepreneurship, and innovation. So she's going to bring an interesting perspective on the intersection of biology, computing, business. Ewelina, welcome to the show.
Thank you so much.
So tell us what inspired your transition from neuroscience research into entrepreneurship and business development?
So I was always curious when, I dreamed about being scientist, but I knew that it will not be only this. And I always was curious about what is outside academia, what is in the real world. So actually, when I was still a researcher, I was exploring and thinking, what can I do? And after many trial errors and some networking, I figured out that I could work with the small companies, I could work with startups, and because they offer a lot of flexibility, and it's also interesting. It's also usually a lot of learning, because when you work with a small company, you can do a lot of jobs at the same time. So I went in that direction. I started to work with startups. I get fascinated by this, how you can use technical knowledge, not only to generate a scientific paper, but also how to change the world. And also I become fascinated with artificial intelligence, because this is a very powerful way of changing the world. And I started to work on commercial applications of AI. And yes, and this is how it started. I transitioned to the commercial side of engineering.
Interesting. Looking back at your PhD journey, what was the most important lessons that still guide your work today?
Well, I think what is very important is that you really have to enjoy what you are doing, regardless the results. And it's always good to have this kind of approach that no matter the outcome, that the work which you do will make you a better person. So I think it's a good strategy, because in research, things usually very often don't work, but you become more skilled and you get wiser, hopefully. So this is important. And I try to keep this rule also outside academia, that every work has to somehow improve me no matter the results, because the results don't always depend on you. And many things can happen, but what is important that you get better. So you either learn something, or that you can become better person through your work. So I think that's something what I did during my PhD, and I try to continue with that.
Amazing. Yeah, I think that self-improvement is core on entrepreneurship, right? And interesting that it's also core on research as well.
Yes, definitely. In every actually risky project where you cannot guarantee the results, because things often don't work. And you sometimes spend months on some experiments and they don't work.
Right. I mean, you can do everything right and still fails, right? But that should not be considered a failure. Rather,
no, because you can learn something
outcome.
Or try to, so that you are better after doing the work than before. So that's important. You yourself,
Right.
you are better, at least.
Right. So how did you first encounter this idea of bio-inspired or wetware computing?
So I was in London at that time, and I was going to many conferences to learn about artificial intelligence. And I met FinalSpark founders at one of the conferences in London. And I was very impressed, because they are engineers. They are actually trained in signal processing. They have PhD in signal processing. So this is totally different. It's not biology, it's mathematics, actually. So I was fascinated that they transition to totally new field. And also for me personally, it's perfect combination, because it's combination of neuroscience, what I know from my research, and engineering, what I know from the commercial work. So this is very cool combination.
Interesting. So at FinalSpark, you're working on building computers powered by living neurons, right?
Yes, absolutely. So we want to build processors, so that instead of GPUs or CPUs, we use living neurons as a processor in the new type of computer.
Amazing. Can you explain us a little bit how that technology works?
So at the moment, we have prototypes, so it's still the real computer doesn't work yet, you could say, but we have done a lot of work in prototyping. And basically, what we do, we make round structures of neurons, which consist of around 10,000 neurons each. And these are called neurospheres, or you can also call them organoids. And these round 3D structures we put on the electrodes, usually on the 8. You can see this on our website, FinalSpark.com. There is a section live, and you can see how it looks, how these electrodes looks. Usually neurons you cannot really see with the naked eye because they are semi-transparent. And what you can see are the signals also on the website. So we measure constantly 24 hours per day, 7 days a week. We can measure signals from neurons, and some of them are also visible on the website. And the whole idea is because neurons in our brain, they generate electric activity through some movement of chemical ions through the membrane. So they generate spikes, so-called electrical activity, and they also can be affected by many chemicals. For example, neurotransmitters such as dopamine or serotonin. And in the lab, we try to use this to program them. So we send them electrical signals through electrodes, or also we can release to the medium, to the liquid in which neurons are placed. We can release some chemicals, for example, dopamine, and this also can change the behavior of neurons. So we try to program them, and the objective is so that they can process information.
Right. I remember that at one point they were talking about mechanical computers, right, which is something that was the past for calculating ballistic trajectories and things like that, but basically were based on gears, right, and
yes,
other mechanisms, right, which at that point was faster than binary computers, right?
Yes.
Now binary computers went up in processing power, but then there has been a renewed interest in these mechanical computers, or basically they're designed for a very specific use case, right?
Yes.
So I'm wondering if something similar is happening with neurons, right? Where you're basically being able to build a specific type of computer for a specific use case that is going to be providing much more efficiency and performance compared to binary computers, right?
No, actually not really, because we are aiming very ambitiously on general computing. So we would like to make a general computing device, and we think that generally the tasks which are today done on digital neurons would be great to be done in living computers. For example, generative AI, because it costs a lot of energy today, and actually generative AI, like solving some complex problems or generating ideas, could be a great task for living neurons.
Okay, so the benefit of the neural computers would be for generative AI, right?
Yes, that's what we have in mind, although it's very speculative at this stage because we are very early stage. So for now, for example, we managed to store 1 bit of information in our neurons. So, just to give you an idea, that is very early stage. And for now, we have some assumptions, but of course, they have to be tested in practice.
Right. Well, I think that's the case when you're on the edge of innovation, right? You're taking additional risks, and it's hard to make any promises in the future, right? Because there's so many unknowns, right?
Yes, I mean, we have, of course, a timeline, we have plans, very specific for the next 10 years. But, it's always a risk, and for sure many things will change, and I'm sure we also will have some unexpected results. So yes, so this is always... it's hard to say what exactly will happen.
Right. So the expectations that you have is that the living neuron computers are going to be able to be faster for generative AI, or what are the benefits that you're planning to achieve with living neuron computers?
Energy efficiency, because neurons are 1 million times more energy efficient than digital, so that's the main thing. They are also easy to scale, very easy to produce large amounts of neurons. And we know for sure they work, because this is why we can talk today, because our neurons are processing information. So we definitely don't expect them to be faster than digital, because actually when we look at the human brain, we can have already some ideas about where neurons are good, where not. So we can say for sure that computers have much more memory. They are much faster for repetitive tasks. But for complex tasks, they use a lot of energy and are not very efficient. And this is in contrary to the human brain. So we expect that neurons in our bioprocessor will be great for this kind of tasks for which human brain is good.
Interesting. Would you be able to do parallelization with the neurons as I understand that's something similar to what happens in the brain, right? Where the brain can have bifurcations on the neuron path and basically can solve complex issues in parallel compared to a single core computer, right?
Yes, actually, living neurons, they can do a lot of stuff at the same time, and also they have much more connection between each other. Also, a lot of recurrent connections, which are possible in digital, but they are much more difficult. So, brain is, we can say, much more connected than digital neural networks.
Right. So, what are the main challenges of developing this type of computers?
So actually there are many challenges because neurons are processing information in totally different way than digital computers. So we have to figure out everything from the scratch again. And this is really difficult because nobody really knows how neurons encode information. We know a lot about processing. We know that they produce spikes, electrical spikes. We know that they encode information in time and space. So it matters when they are spiking and where in the brain. But we don't really know what does it mean. So we cannot read really human thinking today, but that's kind of necessary. We have to be able to somehow translate these signals into some specific knowledge, and this is very, very difficult. And this is why we have our lab fully automated. So basically running experiments in the lab at FinalSpark means that you write a script in programming language, Python, and everything happens automatically because we need so many experiments, such a huge volume, and that's much more efficient. So it's very, very difficult because nobody really knows how to program neurons, and you actually need a new framework, some kind of like mathematical framework to describe how neurons encode and process information and how we should code them, how we should program them. So like kind of new programming language, totally new. So for now, you write code in Python. Of course, we still use a digital interface, and then everything is translated into machinist behavior. So for example, you can code in Python that you will send some electrical spike or maybe some dopamine to the neuron, and then it will happen in the hardware. So you programmatically can change the hardware and things will happen. But we still don't have perfect formula on how to interact with neurons to get some meaningful interactions.
Right, I see. So when you're saying that you don't have a meaningful interactions, are you also referring to the non-deterministic nature of the outcomes? Or that's a different topic?
I think that that's not really clear in science, whether brain is deterministic or not deterministic. I think it's for sure a lot of chaotic and we cannot predict. It's not predictable, let's say. But whether it's deterministic, I'm not sure it's really consensus in the neuroscience. And you are going in the right direction because brain is generally messy. So if we talk about this, it's very messy and it's not stable. This is extremely difficult because computers are a stable system. So you put some input, you get output, and things are working the same way every day. But brain is plastic. So also our brain and the neurons in vitro, they are plastic. So that means they might change over a few days. So for example, today you do one experiment, you put some input, you get output, and tomorrow you can get a different output for the same input. So this is actually extremely difficult, that it's unstable. And for us, for today, it's not possible to predict always the answer. So when we send some stimulation, we don't always know what we get back. So it's hard to predict. So that's why it's so difficult also.
Interesting. Yeah, as far as I understand, we don't have a really good understanding of how the innards of our brain actually works, right?
Absolutely not.
We have some hypothesis, but
Yes.
we have not been able to understand the innards, right?
Yes, absolutely. And especially the cortex, there is a lot of signals and we don't really, for us, it's just a mess in the signals.
Right. And I understand that part of it is the complexity of the chemistry process, but also, and I think you mentioned this, the amount of neurons that our brain has, right? That makes it much more complicated
Yes.
to process and understand the data that we could even collect, right?
Yes.
Interesting. So you mentioned that biocomputer could fit in the future as of AI and data processing, right? Are you thinking of other use cases for the future?
No, we are actually focusing on AI because it's using exponentially increasing amount of energy. And we think that biocomputing can solve this problem because it's so much more, 1 million times more energy efficient. So
Wow.
we try to make solution for the increasing energy consumption by AI.
Right. I see. And does the neuron, maybe this is too early to say, but does the neuron computers suffer degradation, unlike a silicon chip should not degrade over the next 10, 20 years, whatever. They become obsolete eventually, right? But working with a living being, living neuron, then I think that could be the case, right?
Yes, this is very important concern actually, and we put a lot of effort to keep neurons alive for a long time. In the laboratory conditions, especially when you put neurons on the electrodes, it's very difficult. So far we managed to keep them alive on the electrodes for 3 months, which is really a lot for industry standards. We also have some patents for this, for the solutions related. And of course, we want to still increase this time. So in the future, we hope we can keep them alive for years. We know from nature, neurons can live even 100 years because in our heads, they usually don't divide. When we are adults, we have most of the neurons the same. Only in some regions, there might be some new neurons formation, but in most of the brain, we have the same neurons for our lifetime. So we know they can live long, but in the lab, it's much harder because you have to actually to try to mimic the conditions of the human body as close as possible. And every slightest deviation is actually problematic. So that's why it's so difficult to keep them alive for a long time. But we definitely plan to extend this.
Interesting. I thought that the neurons would also go through a regeneration process like the rest of our body, the cells that they regenerate, right?
No, actually, that's a big problem also for humans, because when we have some brain injury, it's very difficult. That's why brain actually has different solutions, because it can rewire. So let's say if we lose some part of the brain, then let's say other parts of the brain can take over the same task. But of course, this is to some limit. And this is actually a big problem also in the neurodegenerative diseases, that usually when you have, neurons, you lose them over your time. So that's really something to worry and care about. Otherwise, yes, we can get slow degradation and cognitive decline over time.
Interesting. Yeah. I mean, I knew about the cognitive decline, but I haven't made that connection. Very interesting.
No, actually, there are many reasons. Yeah, but usually over age, yes, neurons are dying. Just yes, some part, and sometimes you have imaging studies when you can look, the change in the volume of the brain, and especially if people are sick, like if they have neurodegenerative, but even just aging can just make the shrink the brain. That's why it's so
Right.
difficult to keep cognitive abilities for a long time.
Right. We were able to, throughout the years, generations, live longer and
Yes.
keep our bodies, healthier. But now we're getting to the limit of I mean, the body could be healthy, but our cognitive function is not there anymore, right?
Yes, this is big problem. Although I'm sure there are solutions for that. It's for now, at least, a lot in the lifestyle. Nobody yet has a pill, but of course, researchers are working on longevity solutions. So maybe one day we just can drink some elixir and then we get younger.
Wouldn't that be nice?
I know actually there are a lot of research in longevity to try to mimic some of the things related to healthy lifestyle. So yeah, there is a chance for the future, but for now, only the lifestyle.
Right. Also, I don't remember what I read about this, but anyway, I'll try to explain it the best I can. But one of the things that gives humanity hope is that new generations, new babies, are born blank, basically, right? that means that there's hope for change. There's hope for new generations to take new cultures and understand new concepts, right? But the older we as humans we get, right, the more experience we get, the more we tend towards the bitterness and because we maybe not bitterness, but we lose this sense of exploration and awe for new things, right?
Mm-hmm.
A new baby can be surprised by a light that can turn on and off, or a new toy, right? And we lose that feeling the older we get, because we already seen it, we already done it. So there's nothing more that surprises anymore, right? So we kind of lose this concept of joy, right? So if it was the other way around, if new kids would born with the collective memory of the parents, and we as humanity or will be lost, right? Because it will tend towards that bitterness and and lose of interesting interest for anything, right?
Mm-hmm.
So your work on PhD, you produce more than 20 peer-reviewed papers, right? Which publications you feel represents your contributions to neuroscience the most?
I think that my PhD research, because I had very nice topic actually. I investigated the effect of nutrition on brain inflammation, and that actually was very helpful because I changed my view on my groceries at that time, because it was about how nutrients can affect brain. I read a lot of papers about this, and I realized how important it is to eat unprocessed food and try to make the diet anti-inflammatory, because it can prevent some diseases and also can help healing. So healing the brain. So yes, I appreciated the importance of this. And what I studied, some combinations of nutrients in vitro on microglia. So microglia cells are kind of like immune cells of the brain. So I was researching how nutrients can change the activity of microglia so that they are not so inflamed. And I also did a really cool research in brain imaging, so that I enjoyed a lot. That was my favorite. So I did PET imaging, so it's positron emission tomography. It's such a complicated name, but the idea is that you inject radioactive substance, and the radioactivity is something what is radiating. So you can detect this from the distance. So in this way, when you put a specific substance with the radioactive label to your body, you can basically image the body, some function of the body. So for example, activity of microglia, these immune cells of the brain, you can image this non-invasively just using PET imaging. So that was, I think, my favorite studies. And I researched some effects of nutrients on how you can prevent or how you can help to heal the stroke effects. So that was interesting for me. That was the nicest what I remember.
Interesting. What was your most surprising discovery? You built a hypothesis and then you found completely unexpected results that quite surprised you.
Actually, to be honest, sometimes I think that science in some ways is following common sense. Because, for example, we know for years that diet and exercise is helping you to keep healthy. And now we still have new studies being published to show the results that how diet and exercise can improve your brain function and other things, although you knew this already. So I think very often we test something we are kind of sure about. So surprisingly, there is not so much surprise in academic research, I would say. In many ways, you follow the intuition. And actually, it's still interesting, because there are a lot of ancient stuff which maybe people believed sometimes. And after years, you prove them in the lab. Of course, not always. But I would say a lot of stuff are just confirming your common sense about healthy lifestyle.
Right. The grandmother giving you advice that was passed on generation and generations that
Yes.
then
After you prove this in the lab, that is indeed true. And yeah, but I would
right.
say a lot of stuff about the brain. Of course, there are some details about how exactly, which chemicals exactly are working in the brain. But I'm not sure it can be so much surprising, because usually, in the science, I think you make also some incremental little steps forward. So you usually base on some previous knowledge. So you make little step forwards. I think it's not that surprising. Of course, sometimes you have such a surprise, like when, I don't know, penicillin was discovered. Sometimes you have some serendipity, maybe, but in many ways, it's often also you get results you expected. So maybe
Right.
what can be, surprising is when you plan some study and it doesn't work and you don't get any results. That can be surprise. That sometimes can be surprise. That means
Right.
that you either had, there are two options actually for this. You either had the wrong hypothesis or you executed the experiment wrong. So you never know which one.
Oh, I see. I see. And how do you validate the execution of your experiment? Through peer reviewers?
Oh, yeah. Well, actually, first inside the lab on your own, you always have to repeat the experiments. I mean, let's say at least 3 times. It depends on what you do, but let's say at least 3 times. Also at FinalSpark, if we said we'd have done something, that means we repeat it several times and we really get reproducible results. Because especially in biology, it's very easy to get some nice results once, but then after you don't get it anymore. So it's very important reproducibility. It's actually a huge problem in biomedical research because, a lot of papers are not reproducible in other labs. But I think this can be reason also for that there are many little details, that to do an experiment good, you need to take care about many, many little details. And I think that makes also reproducibility more difficult because someone has done this, they were expert in the field, and for others might be difficult because they just don't know how to perfectly execute this experiment. So it's very difficult. But generally, let's say reproducibility in biology is a huge challenge. But you always have to have, you have done experiments independently on different days, on different samples. And then you should get, if you get similar results, you do some statistics, and you can usually statistically say if it's significant or not, the difference. And then you say, okay, then you can publish that as a positive result. And of course, in science, it's a huge problem because usually if you have negative results, it's hard to publish. But I think there is some rationale for that. Many people criticize this, but I think it's not so stupid because actually it's very difficult to prove that something does not exist. Because it's easier to prove, if you have positive results, you say, okay, it exists, it's like that. But if you have negative results, doesn't mean it doesn't exist. Maybe you just done something wrong. So I think it's really difficult. I am not so much against it because I know many people criticize this, that negative results are not published. But, many times they can be just failed experiments. So it's hard to say. But yes, you usually try to do your best with statistics, with repetition on your own, and then later you submit to the journal. And yes, and you have what you mentioned is peer reviewed. So that means you have to wait for a few weeks or maybe a few months so that other people read your paper and maybe they give comments and they say if it can be published or not. So that's
Right.
how it goes usually.
Yeah, it's very difficult to prove a negative, right? And
Yes,
that's
exactly.
a
And some people
challenge.
criticize this, but actually I think many times it can be just failed experiments. So yeah, the fact that you don't get results doesn't mean it doesn't exist. So yeah, very
Right.
hard.
Right. And then the other issue is getting into public publications on a science newspaper. That's also challenging, right? Because you have to go through the peer reviewing process and getting all there until it can actually be published, right? Which
Yes, it's
I
always a
can
long. Mhm.
I can understand I can understand the reasoning around that, right? Then anybody could publish anything, and then the value of the noise start to increase, and then the value of those publications, of those papers, kind of diminish, right?
Yes, absolutely. I think peer review is very important because yes, I think it's really important. Yeah, it takes a long time, but also researchers actually are doing this for free most of the time. So when you are academic, you want to become professor, then usually you are volunteering for reviewing the articles and you practically do this for free. So it's a lot of work. So yeah, I think that's maybe the reason why it takes so much time.
You mentioned a little bit before about using imaging technologies. What drew you to combine it with neuroscience?
I think imaging is very powerful for neuroscience. You can measure in vivo what happens, not in the sliced brain, but in the real living brain. So yeah, I think it's very powerful and it allows you to do research also in humans. That's very important because you you don't do only animal research, but then you can translate easily to human because you basically can do the same method. So that's actually easier, because what is the objective of all the medical studies most of the time is to find out how to cure human brain. So that's always important, that things are easy to translate. So yes, and it's very non-invasive. It's, let's say, painless method to study, what happens. And it's used in neuroscience, also in psychology, in any actually research which are connected which are about human brain. It can be used to measure activities. And I think it helps us to understand how brain works. Now there are even some kind of attempts to kind of read human thinking, but of course it's attempt only, because you can do only classification. So for example, if you give to the subject like 10 different images or sounds, music, then you can classify which one the subject is thinking about based on imaging. But only if you know previously which music or which, so it's not like totally reading your brain, it can guess which one you're thinking about from a predefined list. So yeah, that's
Right.
such interesting stuff. So yeah, I think it's
Is
cool, and it's used more and more, and not only in research, also a lot in diagnostic, because also it's used a lot to, for example, detect tumor and stuff like that.
right. Right. Yeah. I understand that what they're doing is kind of blindly compare the images, right? The scanning images, right? It's like, I don't understand what's going on, but if they look the same,
Yes,
it
in
kind of
many
goes
ways
that
it's
way.
like that. When you do classification, yes, then you can just say that when you think about each music piece, then your brain activity is a bit different, and you can compare. So sometimes you know what is going on, sometimes you don't really know everything. So yes, sometimes you blindly compare. Sometimes you know exactly what you're looking for, especially for diagnostic. When you diagnose, then let's say you should know what you're looking for. But if for research, sometimes you don't really know exactly.
Right. Right. How does the neuroscience compare when you work with animals versus humans? Is there a big difference? The easily to translate is something happens in animals, then we can safely assume something similar happens in humans. Is that translation exist or it doesn't?
So I always work with animals. So I think it's maybe a little bit easier. I think with humans, you have much more bureaucracy. This then becomes a lot of paperwork when you work with human studies, not so much experimenting. And you collect some little data, but usually it's a lot of stuff, a lot of ethical approvals, because the human studies are the hardest. Generally, this is big problem in biomedical research. It's not easy to translate. You might have some drugs stuff working in animals, and later in humans they don't have to work the same way, because of course animals, they have different enzymes, different metabolism, different doses. There are a lot of difference. So yes, it's just a model, but many things don't work the same way. So it's always a risk when you test something in the humans that it might not work.
Right. Right. I was reading about full body scan, right? And one of the reasons people were suggesting not to do those type of full body scans was there's always something wrong with your body, right? So that will trigger false diagnostics, right? Or would scare people, right? So even though it could be a preventive mechanism, right, to prevent the early detection of any sickness, but it also opens the door to all these false negative issues. I'm wondering how much of that situation, like every human body is different, how much of that actually would affect your neuroscience research, right? Because at the end of the day, you need something repeatable. And if you have a non-deterministic human body subject, if you like, right, then how can you make that distinction between, is this a variation of the patient, or is this a variation of the actual sense that you're trying to prove?
Actually, I don't agree, to be honest, with this full body scan. I think even if you have false positive results, you can always double check. And I think it's better to know than not to know, especially many cancers, it's so much important to have early diagnostic. So I generally think that it's good to do preventive studies. The only problem is that there are many ways of imaging. So if you do full body scan, which techniques do you use? Because there are so many different techniques and they all show something different. So actually, when you do this full body scan, it depends a lot which technique you do, and you cannot always see everything. So I think it's important to use maybe, I'm not sure it's necessary, definitely not necessary to do PET for diagnostic, just maybe it's enough to do MRI, but that's good for brain. But for the rest of the body, there are other techniques which are cheaper, for example, ultrasound. So I think it's important because I know there is this marketing of doing full body MRI maybe, but I think that's a little bit marketing because at the end, there are many techniques and they all show something different. So if you would like to be really sure, then you would have to use different scans. So I think that sometimes is marketing, but generally it's good to do diagnostic because the earlier you know, anything is wrong. Even if I think it's better to get false positive than not to know when you're sick and then, get terminal. So that's my opinion at least. And when you do the studies, you always have to have different subject. And that's kind of like maybe kind of helping you to solve this problem. Because, okay, you can have false positive maybe with one subject, but if you repeat a lot, that's why repetition is so important in any kind of bio studies, that you have to always repeat. And if you get results consistently, then you can say you have something. Otherwise, it can be just no coincidence, indeed.
Right. Yeah. I still remember on my statistics class where they would talk about, a sample that is statistically representative, right? Which is
Yes,
basically
always.
with volume, then you can get all the specific samples deviances out of the actual metric, right?
Yes,
Yeah.
absolutely.
And yeah, I agree with you on the full body scan. I've seen the same behavior with the Apple Watch and these, health trackers where people get obsessed about the numbers and the metrics, and when the Apple Watch filling the rings. And I think it's important to have that information and know rather than not knowing. But then what do you do with that information, right? Do you take it as an absolute, right? Then that do you overreact for any, I haven't filled in the rings for the last 2 days or whatever, right? So I don't think we should react to those metrics like that, because that's not healthy. But being able to know, right, and being able to be consistent on improving the numbers towards the goals we want to do, I think that's much better than just obsessing with the metrics.
Absolutely.
Yeah. And I agree with you also on the, it's better to have a false positive and work it out than not, right? Yeah. I also seen that when you're doing imaging, it also can define the level of detail that you're getting. So if you're doing a full body scan, you could get a low resolution and then focus on the parts that you want to focus the most, right?
Yes.
Which, that could also affect the results.
Yes, absolutely.
So you co-author on gene imaging and CAR T cell therapy, right? What was the biggest insights for those studies?
CAR T cells, okay, this is on cancer imaging. Actually, I continued in London as a postdoc in cancer imaging. And CAR T cells is very cool, a new technology on which we were working on preclinical studies a few years ago, but now I see a lot of studies in humans also, and they are effective. So this is when you use the genes from your own cancer, kind of, from your own body. You use the genes to modify T cells. And then these modified T cells you inject to the human body, to the patient, and they should kill specifically the tumor in this patient. So this is very specific. This is example of personalized medicine, where you had a treatment exactly for one patient. So this is most personalized possible, that it gets treatment specifically for this one. And I think in cancer is often necessary because every cancer is different. This is actually terrible, the variety, because it's genetic mutation. So mutation is random. So that's why what comes out is always a little bit different. So that's why it's so much important to have personalized medicine. And also it's important for any type of metastasis, because these CAR T cells, they are flowing in the body. So they can go anywhere. So they can also help with the tumors which are spread over the body, which is a huge problem. And actually, in many cases, they are terminal. Because if you have metastases, okay, they can be everywhere. You cannot eliminate the cancer from the body. But with CAR T cells, there is a hope for doing even this. So yeah, that was interesting. I was doing a preclinical on mice just to prove that CAR T cells can arrive to the tumor. So we were using imaging to show it.
Wow, very interesting.
Yes, just to confirm that they can really arrive where they should.
Right. So basically, you can attack on a cell level. Yeah, because that's always the issue with cell tissue, right? With cancer tissue, right? If you cut too little, then you're leaving unhealthy tissue. But if you cut too much, you're cutting healthy tissue, right?
Yes, absolutely. But with CAR T cells, it's kind of like super precision because this is biology. So your own T cells are fighting. So this is much more precise. And also it can go everywhere in the body. So even if the tumor cells are in different part of the body, it can still arrive there.
Yeah, and also it's less invasive, more precise. You wrote a paper about the open and remotely accessible neuro platform.
Yes, this is
right?
very relevant paper for our discussion today. Yeah? This is actually about FinalSpark. This is the latest publication, and it's actually very successful because we published this in Frontiers, in peer-reviewed journal. It was accepted after the first. Sometimes you have to try many different journals, but here we didn't have to. We submitted to Frontiers to the special edition, and we were accepted. And what is very interesting about this paper that it's in top 1% of all the papers read in this journal. So I'm sure it's in big part due to our efforts because we try to promote this, but we are very happy for this. And this paper is not really experiment, it's more description of what we have in the lab because our lab is available remotely for researchers all over the world. So basically you can access through internet browser and you can do experiments, collect data, analyze the data, do everything, do full experiment in our lab without ever entering in the lab. So you can do this from any place in the world. And we try to describe as much in details what we exactly have because people often come to us, of course, with expectations which are not possible to fulfill. So we want that all the engineers or scientists who would like to collaborate with us, that they know exactly what we have in the lab. What can we do and what is possible today so that we have everything automated, 24 hours per day, 7 days per week. You can run experiments, send in electrical signals to neurons, you can receive the response. And yes, it's very robust tool for biocomputing, for prototyping, for learning how we can program neurons.
Interesting. I would love to learn more about that. I find it fascinating. I mean, I'm a software engineer, so being able to automate, living neurons research, I think that's fascinating.
Thank you so much. We are very happy to hear that we get messages from people from everywhere in the world, and there is a lot of excitement about this project.
Right. Yeah, and the I'm just thinking about the possibilities, because now you can have a globally distributed team of scientists working on the research, and as you said, 24/7, right? So that's another huge benefit.
Yes, and it also shows that our research is scalable because you can easily incorporate new groups in the lab without increasing the floor space so much.
Right, and repeatable because now you
Yes.
can repeat it as many times. That's
Yes.
amazing. So how do you envision researchers around the world are going to use this remote biocomputing resource?
So we have already 10 universities which are using our lab for free, and they have some ideas. For example, about how to research connectivity inside neurospheres because it's 3D structures and we have signals only from the surface, but there are some mathematical methods to figure out what is inside. And what is also surprising for us, nice surprise and side effect, which we didn't expect, is that we have first commercial clients because there are people who reach out to us, companies, individuals, startups who are working with us, and they pay us to get subscription, to get access to the lab, and try to do some R&D on our neurons.
Right.
So sometimes, Sometimes there are startups who have some novel ideas related to biocomputing, and sometimes there are big corporations who have R&D teams, and these R&D teams want to know what is on the edge of science, and they like to try out the new technologies, even if they don't work yet, but they want to know what will be possible in a few years and get familiar with it.
Right. What were the technical or ethical considerations you had to address while publishing on living neuron-based computing?
So, well, technical and ethical, they are the two different things. So technical a lot. There is huge amount of stuff you have to do to make things possible, and we are still working on improving our lab. And for ethical, we get sometimes questions from the public because project become popular. So people are starting to ask questions, and this is why we reach out to philosophers who are working on technologies so that they help us to answer some questions related to biocomputing, because of course we want that our work is accepted by society. So if there is anything we have to do, we will be happy to do this to make sure that it's ethical what we do.
Right. Right. So we're running out of time, so I'll ask one last question before we wrap it up. But how do you imagine the future of science, in the next 10, 20 years? And what role do you see living systems as biocomputing you think are going to play?
So I think a lot will change in the near future because of generative AI, because of the LLMs, because they give new, very potent possibilities for automation, and even academic research, experimental research, engineering can be at least partly automated. So I think it's a huge change, and I'm curious how it will work out and how the future will look like. So in around 10 years, we want to build bioservers. So this will be centrally available biocomputers, which you will be able to connect to, like to the cloud computing today. And this will be cheap computational power. So this is what we aim for.
Amazing. Amazing. So before we wrap it up, Ewelina, any last remark you want to share with our audience?
So I encourage anyone to reach out to us if they have any question and to check our website, FinalSpark.com. We also have Discord community, so you are very welcome, everyone, to join. There are a lot of discussions. They're also technical, so I'm sure it's a lot of fun.
Amazing, amazing. I'll check it out. I think what you're doing is amazing, and
Thank
I'm
you.
looking forward to hear more news.
Thank you so much.
Great news about what you're building. Thank you, Ewelina, for being today.
Thank you.